All right. Here we have, it is Friday, August 28th, AP Biology. So today, agenda's coming. We're going to go over some notes on experimental design and then start on a which would a woodlouse prefer assignment. For this week, things to make sure that you have done: the student information form, the syllabus quiz. Some of you did it on Schoology. Those of you that were dual credit had to take the quiz on Canvas. Your pre-quiz on AP Classroom. And then our CER intro activity, having 3 responses on that discussion board. And then the practice FRQ from yesterday. For homework this weekend, this is an AP class. You do have a little bit of homework. 2 things. You need to get some M&Ms and have them ready during class on Monday. We'll be using those M&Ms as part of our activity, and having them in front makes it far easier, or having them in front of you will make it easier for you to follow along. And then some chi-squared notes. Where'd my mouse go? All right. Let's go to our Schoology page. And here on Friday, you see a page that says chi-squared notes. So in those notes, there are a few different videos you can watch. You can watch all 3, you can watch 1, you can watch 2, as well as some reading material so that you are prepared and familiar with chi-squared before we talk about it in class and practice using it. So over the weekend, get yourself some M&Ms and study up on chi-square a little bit. All right, now for today, our experimental design notes. Let's make this nice and big. All right, a lot of this should be review. Our first slide here is about our variables, independent and dependent variables. The independent variable is the variable changed by the scientist. It affects the dependent, and when we're graphing, it typically goes on the x-axis. Dependent variable is a variable that's measured, depends on the independent, usually goes on the y. We think back to yesterday's FRQ with the rats and the alcohol. The independent variable was the type of fluid. The scientists changed up whether they had water, 1% alcohol, or 5% alcohol. The dependent was the amount of urine produced. That's what was being measured. Constants versus controls. Constants are things that remain the same for all group, whereas the control or control group is a group that's not manipulated, not changed by the independent variable. So in the case of our rats from yesterday, the group that was given water to drink, that would be the control group. That is the normal group. The groups that were manipulated, the 1% alcohol and the 5% alcohol, we could call those experimental groups. There could have been more. There could have been a group that had 10% alcohol. Next, and for some of you, this may be new. This is not something we usually talk about, at least in pre-AP biology. Is there are different types of control groups. Negative control is what we usually think of or we're referring to when we talk about a control group. This is the group that's not exposed to the experimental treatment or any other treatment that's expected to have an effect. So our rats with water, they would be specifically a negative control. In the rat experiment yesterday, there was only a negative control. Some experiments will have a positive control. This is a control group that is not exposed to the experimental treatment, but that is exposed to some other treatment known to produce the expected effect. you see this happens a lot actually in medical studies, looking at new drugs and their effectiveness. We'll have a negative group that gets a placebo. We don't expect their illness to get better. We have a positive group that has the current existing drug or medication. We expect their improvement. And then our experimental groups would be then the people who got the new drug. We want to see if it works as well as the old. Again, most experiments only have one. Some experiments will have the positive and the negative control. Next, hypothesis versus prediction. And you can blame former teachers, probably including myself, for oversimplifying a hypothesis. Most of what you know as a hypothesis and what the RAT FRQ considered a hypothesis is actually a prediction. A prediction is a statement of what you will think happened. So in the RAT FRQ, if you said rats with alcohol will produce more urine, you got your point. That was really a prediction. Oftentimes, in other science classes, you're probably asked to write things in an if-then statement. That is perfectly fine. For an if-then statement to be a full, complete hypothesis, though, it needs to be if-then because. If we look over at hypothesis, in a hypothesis, you should use accurate scientific concepts to explain your prediction. So you have to say that because or that why. So in the rats, for it to be a complete hypothesis, you would have needed to explain why you thought urine production would increase. Now, the rubric did not expect you to do that. Later on in class, when we are writing hypotheses for lab reports, I will expect you to put in that because. And when we look at our examples today, we're going to put in a because. Data collection and observations. So data tables will be used for your numeric data or your quantitative data. So the rat scientists measured in milliliters. They had the numbers for how much urine was produced. Observations tend to be more descriptive, qualitative data, describing things what would happen. So in our rat experiment, they did not give us really much qualitative data. Qualitative data might have been observations such as the rats that were drinking the alcohol. Maybe they seemed sluggish or weren't walking as well compared to the rats that just had water. And then finally, here it is, CER again, which we talked a lot about yesterday. This is what we'll use at the end of a lab when we do our data analysis and discussing our conclusion and supporting, arguing why your conclusion is correct. So here we have a question: how does the temperature affect the respiration rate of a goldfish? Your claim is your answer. Higher temperatures increase respiration rate. Evidence. So here in the evidence, we have some sentences describing the data. And then reasoning, going further. So providing that scientific principle, saying that oxygen is required for respiration. The faster a fish moves the gills, the more oxygen the fish is able to bring in to maintain that respiration rate. Okay, let's go look at your assignment again. Not again, your assignment for today, now that we're done with our notes. And that is the which would a woodlouse prefer data nugget. There should be a PDF attached. Let me fix that. We can go ahead and open up our document that we'll need. All right, sorry. So here's our data nugget. Has two parts. This is part one. Part one is all we're going to worry about today. Reading the background information, which I just added to Schoology. We'll go take a look at that. Finding the hypothesis, determining our independent and dependent variables, and then you will need to create a graph. If you know how to do one digitally, that's great. I would recommend do it on a sheet of paper, sketch it out. Then you can take a picture and send it to yourself. To insert a picture, you just come up to insert, image, and then upload it from your camera or from your laptop, your computer. All right, let's see, let's refresh this. Here we are. Now, here's our PDF. Maybe. There it comes. All right. All right, so in our PDF, you're going to read through this. When you find the hypothesis, I just want you to copy and paste it directly into that data nugget page. But this is all about a scientist by the name of Nora and some research she did on wood lice, is the plural. Here in Texas, we usually call them either pill bugs or roly-polies, but same thing. Read through her experiment. There's even a few pictures, and then you'll be graphing the data. All right, that is it for today.